This media was gratuitously produced by Clear Contributions Audio Video. The intellectual property contained within belongs to Dr. Richard Gardner and Garland Science. Clear Contributions. Let's have clarity.
What I want to do is start by introducing these papers. And again, some of them are recycled from last year, but for the last couple of days of class to do these presentations. And I've been doing this every year in cell biology and genetics as well. And I don't know what's going to happen in genetics next semester, because we got like double the number of people we had before. So I don't know if we can keep doing this, but I've got 6 of them. So I'm kind of envisioning 4 groups of 3 people and 2 groups of 2. That makes 16 altogether, and that's what we got here. And the hard thing is to try to figure this out without being socially awkward and like, you know what I'm saying here. But let me just tell you what the papers are, and I've got a little sign-up sheet, so if you kind of have an idea of who you might want to present with, and if we get groups of 3 and we end up with 4 people left over, we'll split that and put the 2, or if you, we don't have time probably for everybody to do a group of just 2. And of course, it's not divisible by 3. 4 people is kind of too many, which is why I don't know what we're going to do next semester, but I haven't figured that out yet. One of them is slightly longer than the others. These are all shorter papers. I tried to find more or less shorter papers. Sometimes they're 10 to 15 pages, but I've got ones that are more like 5 to 6 in most cases. This one's about anthrax toxin, and it's how it's kind of endocytosed into the cell. This one, we haven't even talked about the cell cycle yet, but a couple of cell cycle kind of papers. This is the mitotic checkpoint, which basically is that the cell looks before it goes into anaphase, before it tries to split its chromosomes and separate the nucleus. Before that happens, it makes sure that all the chromosomes are attached to the spindle. And if one of them is not attached, then that's bad. So then it stops and waits till it's attached. And sort of how does that work? What's the mechanism for recognizing that it's not all attached correctly and then for pausing it? So that's kind of what this one's about. This next one is about regeneration and appendage regeneration in mice. They think they've got a gene that's normally good, but if they knock it out, you can regenerate, like they put a hole in their ears or something like this and it regenerates better. And it's anyway. Yeah, here's the hole right here. And then we've got one about Golgi stacking. This is one where they've gone in and mucked around with the Golgi and actually put some of the proteins that are normally on the Golgi on the little individual stacks to kind of hold them all together onto other organelles, and they put them on mitochondria, and the kind of mitochondria then sort of stacks up with the Golgi. It's kind of a interesting bit of engineering. See, that's 1, 2, 3, 4. Here we have a MAP kinase cascade. This is the one I kind of put up on the board before. This is causing the cells to change their shape when they're about to shmoo, or when they start to divide in a different pattern where the cells all stick together in a chain. And then finally we have a flippase paper. A protein kinase network regulates function of flippases. So there's sort of a pathway where this thing activates, this thing activates, this thing activates, this thing, one of those kind of things. They're all pretty technically complex. So again, the idea is that you would read it, and these always take more than one reading. I mean, even someone that's been in the field for a long time, you never can just read it one time and get it all. You want to go back a couple of times at least, look at the figures, and then see what you can get out of it. And then during the last week, right after Thanksgiving, I'll try to meet with each group for about half an hour, and we'll go over the paper. But before that time, you'll have read the paper, because the first thing I ask is, did you read the paper? And so this is mandatory extra credit. Everybody gets free points for it, but you got to earn your free points, if that makes any sense. By actually reading it and seeing how it goes, and there's going to be stuff you don't get. I mean, that's just how it is. So don't panic about it. Just try your best and see what you can do. And it's great practice reading papers like this. It's not like anything else you've ever read, probably, if you haven't read one of these before. Does anyone have right now a clue of who they might want to work with and this paper they might want to discuss? Let's see. 1, 2, 3, 4, 5, 6. I feel like I'm missing one. In fact, if anyone wants to make any deals here, I don't know if that's helpful or not, or do you want to just after class or whatever on the hallways while you're eating lunch or whatever. I want to get this figured out this week so that, because then next week we'll have, how many people, by the way, I hate to even ask this question, but how many people are not going to be here next Monday? So they're not going to be here next Monday. Next Monday is Thanksgiving is next week, and then, I'm going to be here anyway. I'm just, we have a test scheduled for next Monday. So if you plan on leaving early, it'd be nice if we could do it on like Friday, and I haven't even written it yet, but that's my goal for today. Half the campus usually leaves let's take off the whole week and let's take off in a few extra days. They'll leave on Wednesday, this coming Wednesday, just because they might as well, because they got a cheaper fare, and then gone the whole next week and missed, But it'd be nice before people leave, you have the paper, and then you can read it over Thanksgiving break while you're digesting turkey and whatnot, and then we'll get back. And as soon as we get back from Thanksgiving, we have a really only a week left, and then the last week. So it's we're really coming up toward the end here. Yeah. Does anyone know yet? I'll tell you what. Let me. Now, of course, the trouble is everybody's gonna. I'll just pass it this way, and it's gonna be first whatever. So think about a second option. None of them are as exciting as I tried to make it out to be. I mean, I tried to make it all exciting, but I mean, they're really cool. But you get into the nitty gritty, and a lot of these have Western blots in them, and you see various protein levels at certain places or not, and they can tell if the protein was there or not. So the idea is then that everybody will have sort of a PowerPoint, or whatever kind of, whatever the software is, I don't care, but you have some kind of presentation we can put on the screen here. And all of these papers, I printed them out, but they're all available online for free. For any one of them, you can get there. There's usually some number that you can type into, but you can always just Google the title. If you just Google the title, you'll be able to find the paper. And then once you get the paper, you can cut and paste figures. And it's not necessary to use every single figure for a presentation. You might not have time to even finish if you do that. So you kind of want to tell the story, the main thing, what the paper was about, a little bit of background, what they did, what the results were. And this is going to be, about a 12-minute or so presentation-ish. Let people ask questions when we're done. So, if we do. And then you don't necessarily have time for every figure. But for the ones you show, you want to be able to kind of have it down, whoever's going to talk about it, kind of what it is. Sometimes people will say, oh, yeah, there's this figure. Yeah, let's see. Oh, can't remember what it's about anymore, but it sure is pretty. You kind of want to have it kind of down, what they did. And yeah. All right, we'll give another couple minutes for this paper to go around, and we'll see what we can come up with. And if we don't all get it done today, then I'm going to put this outside my office on the wall in that little bulletin board, and then people could come by, and I'll put these papers. If you have a group, I printed 3 papers, 3 of each paper. And if you have a group with only 2, then you got an extra paper, but it's easier for me to just print them all out this way. And I'll put those out by the door as well, and you can pick those up. But you want to kind of know who's doing what. So, is that paper still going around somewhere? All right, so it's still somewhere. So while we're waiting for that paper to go around, we had a request to talk more about GTP caps or ATP caps. First off, ATP caps are on which kind of cytoskeletal element? Those are on actin, starts with an A, and GTP caps are on microtubules. Again, when you have the GTP attached to your tubulin or your ATP attached to your actin monomers, that stabilizes the thing, so they're all kind of stuck together. And once your ATP loses its phosphate or GTP loses its phosphate, that's when the end becomes unstable and it unravels. If it's a microtubule, the protofilaments kind of peel off and then they fall apart. If it's an actin, they just fall off. But they're kind of more, they tend to stick together. I mean, the subunits come on and then they'll stick, but the ATP helps them stick better. And if they don't have ATP, then they'll just fall back off again. So once these things lose their phosphates, then it's unstable. It might stay together a little bit, but more likely it's going to just start falling apart. And so the end of it, the new ones coming on, the new units coming on, always have their phosphates on them. So that's why this has a cap. They all have phosphates on this end, but then as more and more new ones start to come on, these have been on longer, and so there's just more chance that they're going to lose their phosphates. So here it's losing their phosphates. And then more of these will lose their phosphates over time. And if they lose them before new ones come on, then there's no cap left. And then it basically disintegrates. And as it's disintegrating, new subunits might join with phosphates, and that will suddenly stabilize it again, and then it can start to grow again. Is everybody more or less good on that idea? The stabilizing caps idea. If you're looking for the easiest one, it's not going to help. They're all hard.
I'm trying to figure out which ones are already done.
Oh, well, let me see here. Oh, let's see. We have the mouse one is this one. So that's done. The Flipase one is now also done, and the Golgi one is done. So that leaves these 3, and they're all good. I mean, they're all interesting. If you pick one, I'll say, good choice, good choice. It's like when you go order something from the restaurant, no matter what you order, the waiter says, oh, that's an excellent choice. You know, as opposed to like, oh, that was a crappy choice. What are you ordering that for? I mean, you know, they don't ever say that. So while they're signing up for this, I'll just tell you one more thing about papers. So when you're in grad school, pretty much is not so much textbook anymore, it's current literature, current stuff. So whatever you're studying, you're now focusing on a very specific, you're working in someone's lab. Is anyone here interested in going to grad school in any kind of field similar to this, as opposed to like medical school? I can't speak much for medical school. If you do that, you get to dissect dead people and stuff. That's about all I know about that. If you go to grad school in any kind of a field that's sort of cell genetics-y kind of, and it's probably true for also environmental ecology kind of stuff. Basically, your first 2 years, you end up doing a lot of classes, and you may have textbooks and all that stuff, but you get more and more starting to read sort of current papers, and they're kind of difficult to read, and you have to get into that. And they're not every program is exactly the same, but this is kind of just a overall kind of how they mostly work. You end up working in a couple of 3 or 4 different labs your first couple of years. They have a lot of places will have these rotations where you kind of set up to work 6 or 8 weeks in someone's lab and then choose someone else. And you have to have sort of mutual agreement with kind of who you're going to work with and if they have space in their lab and whatever. But you kind of figure out who you're interested in, what kind of faculty members have interesting things that you like to do, and you kind of work in their lab. And then at the end of your couple of years, you choose one of them to do your dissertation work in, and it has to be a mutual agreement. If they don't like you, then it's not going to work. So you have to kind of like each other, and you kind of and then you basically spend the rest of your grad school in their lab. And then by this time, the first 2 years, your kind of formal classes are done, and then after 2 years, it's pretty much you go work in a lab all day long, every day, and do your research. And you do that until you're finished, however long that takes. And if your project keeps having disappointing setbacks, which they all do, it'll just take you longer. There's no set time. It's not like medical school where you're going to be there for exactly 4 years and be done. You there and just do your project, and it's pretty much a full-time job. They told me they want me in the lab at least 40 hours a week, if not 50. Don't think about having another job on top of that. Now, if you're in some kind of a molecular genetics kind of field, there's usually money. The professor usually has some kind of grant money, so that pays for your tuition, and it gives you a slight salary, so you can actually not have to have another job. Now, when I was doing this, I got $13,000 a year, which we actually lived on. Which we lived in this dumpy trailer, and it was just dumpy. It was just bad. But they pay your tuition, so you shouldn't have to have student loans. Now, if you're in some other fields, if you go in humanities graduate school, they don't have that kind of money, so you're on your own. You have to teach a whole bunch of classes to earn your money. Sometimes you'll end up having to TA a class because they don't have enough grant money for research. And sometimes they require that anyway. For a year, you have to be a TA. It's a good experience doing that as well. But then usually you end up, because your research, whatever results you get, they're going to basically help your professor. And so that's going to help fund his grants anyway, or her grants. After your first 2 years, usually there's this qualifying exam, and you have to take this exam, and if you don't pass it, they might give you one more chance, and if you don't pass it, they kick you out of the program. So this is a highly stressful situation. You've been there 2 years, and you want to finish finish it all up, and so you have to pass this exam. And then once you pass it, you just basically do your project the rest of the time. And you'll be reading lots of papers from other people in the field, and then the goal is to write some as well when you get stuff done. Generally, and I only have experience where I was a grad student and then where I was a postdoc, but both departments, and I think this is a kind of a largely done thing, but the departments will have sort of a weekly journal club. And in the journal club, someone will get up and present a couple of people will present some articles every week. And everyone's kind of expected to come, all the professors, the grad students. This is all sort of grad student and above, and people working in the labs and whatever, and come and listen, and the whole department gets together. And the way it worked when I was in grad school was that each week a faculty member and a student would present a paper. And I think you got to pick your own paper. If you were a student, they wanted you to talk to a certain designated faculty member first who was in charge of the journal club program, just to make sure you weren't going to make a fool of yourself and make sure you understood the paper. But you get up and present it. And back in the day, it was overhead projectors, but now it's PowerPoint. And you talked, and the whole department, and it was a way that everyone kind of keeps up. No one can read all the current literature coming out, all the papers coming out. No one can keep up. You're supposed to kind of keep up with whatever's in your field and read the new articles coming out so you know what's going on. But no one can keep up with everything. So this way you come to this thing every week and hear about something new and kind of keep up with what's going on, and then a faculty member would present one. The way it worked at UVA in our department was that each month they had a certain topic. So there'd be the first Tuesday of the month, some professor would get up and say, well, we're going to talk about this topic this week, maybe cytoskeleton. And there's 3 new papers that just came out in the last 6 months, and kind of gives a little introduction, remind everybody how cytoskeleton works. And then they'll say, and then next week, so-and-so is going to be presenting the first paper, and then the following week, so-and-so is presenting the next paper, and the following week, so-and-so. And those are all students. So they had kind of organized by topic, and then you hear the next week, and the next week, the student presents these current papers, and then next month they'll have a different topic, and faculty member introduces the topic again, and everyone was kind of expected to show up. I was a postdoc at this time, so I didn't have to do it, because it was basically the faculty member introducing every month, and then the students had to do it, so I didn't have to. And so the idea of kind of presenting these papers is kind of like we're trying to replicate that experience when people get up and present a paper, and we all kind of listen to it and tell us, and you get to say what was good about the paper and what was bad. And if someone did an experiment you didn't really agree with, or you didn't agree with their conclusions, you kind of thought they were overstating it too much, or if they sometimes there's stuff that you just don't agree with the way they interpret things, or maybe they didn't do a really good control they could have done. And maybe they just didn't label their figure very well. Some of these figures have all these multiple little parts to them, like this, and all these, and they just didn't label. It was very confusing. They could have done better. You can trash them. You get up there and say, well, they could have done this better, or whatever. These all have been peer reviewed, so they should have a certain level of quality, but that doesn't make them untouchable. I mean, they can still be problems with them. Peer review process, basically, if you are like a faculty member at a research institution where you're doing research, and editors will send you papers occasionally and say, could you please peer review this paper? And ideally, it's all confidential. They're not supposed to tell anyone else what it is, and it's anonymous. The authors don't know who the reviewers are, but you review the paper, and here's what I liked and didn't like. And it's not so much you're going to correct their grammar, but it's more of like the experiments. I want you to go back and do this experiment again, and I'd like to see better results. I want to see replication. I want to see better controls and whatnot. But typically, a professor, if he has a bunch of people working in his lab, like a bunch of grad students and postdocs and technicians and whatever, lots of times they will copy the paper and invite everyone else to read it, can help them out. And it's a great experience for everyone else to go through there and try to find holes in their thing, And if you can't find any, then you feel like, oh, I didn't do my job very well, So I'm like reading, and if you find something that's wrong with it, good, I found something, then at least the professor will give me a pat on the back that I found whatever. So then they'll have a meeting, and we'll go through there and say, hey, I didn't like this and this and this, and someone else didn't like this. And he's like, thank you. I didn't even notice that. But now that you mention it, you're right. So he puts that in his review, goes off to the editor. And I don't know if this is strictly legal because it's supposed to be just, confidential, but it's a great time for everyone to kind of be able to look at paper that hasn't been published yet and kind of see if you can poke holes in it. So it's a great experience. So if you find things in these papers that have been published and there's little holes in them, that's great too. This is all off the topic of cell biology per se, but it might be useful for kind of how stuff works. All right, so let's talk about the Kirschner-Mitchison paper. I've never met either of them, but when I was at UVA, the guy next door, the professor, he had a lab next door to ours, and he was a new professor. It was like his first job, so he just started there. And he had worked in Mark Kirschner's lab. And Mark Kirschner's a super bigwig. And so the guy that worked next to us, next door, had worked in his lab and never let anybody forget it. Every day it was, when I was in the Kirschner lab, this is how we did it. When I was in the Kirschner lab, well, Mark Kirschner did this, this is your badge of honor that you worked in the Kirschner lab. So Mitchison here was a grad student in his lab back in the 80s. So let's just kind of go down. They knew that microtubules existed, but they didn't really know much about what there was, how it all worked, and dynamic instability. I like this last little bit on the first page, sort of the last sentence. To a young assistant professor, it seemed that the leaders in the field had made major advances on the problem using straightforward biochemical approaches. Although frog eggs would later loom very large in the Kirschner lab for understanding of microtubule assembly and control of the cell cycle, the most immediate approach seemed to be to explore biochemical reconstitution if there was anything left to do. So they kind of thought they understood microtubules pretty well. There was not much left to do. Everyone kind of got everything. At this time, they hadn't yet discovered dynamic instability, and so that was a huge thing, but they thought they'd kind of figured out everything. on the top of page 2, one peculiarity of both tubulin and actin was that both seem to require energy merely to assemble the polymer. Most protein complexes, even very large viruses and ribosomes, assemble without hydrolyzing GTP or ATP. Possible role of energy in the assembly of microtubules always intrigued Kirshner. So what do we know now? Does it really take energy exactly to, so GTP is involved here. Does it take energy to put this together? Or ATP is involved in actin? It's not really that they're providing energy, that the tubulin comes on and it attaches, and it's there it is attached without hydrolyzing anything. It's after it's attached that the hydrolyzation happens. So the GTP and the ATP is not really used for energy to put it together. It kind of assembles by itself. It's used instead as sort of a way to make it unstable once those hydrolyze. So it's kind of a different role of ATP or GTP than sort of just energy. And then it goes on down on this, right above this little figure. Despite early reports that energy was required for assembly, Penningroth and Kirshner were able to demonstrate conclusively that non-hydrolyzable GTP, that means it can't lose its third phosphate, could promote assembly. We now know that hydrolysis follows assembly, and that energy is later used to disassemble a polymer. All right, so then they talked about treadmilling, and they've got a great little picture of treadmilling going on here. Subunit adds to one end preferentially and leaves the other end, and so you kind of see that subunit looks like it's moving backwards, but it's really just staying in the same place while the end gets added and the other end gets taken away. So then we come to the Mitchison part. So now Mitchison's the grad student. What was his what almost happened to Mitchison? Nels got kicked out. And why was that? All right, so they didn't. So you have a committee. So this is the other thing I forgot to mention. After your second year of grad school, you finish all your coursework and you pass your qualifying exams. A lot of times these qualifying is both written and oral. I had to do a written test and then an oral test. The oral test, and everyone does it differently, but the two formats I've heard of most are you have to think about what your research is going to be for the next several years, what your dissertation project is, and think about all the experiments you want to do, and then you have to explain those to your committee, and then your committee quizzes you on it and tries to make sure you actually know why you're doing this, and they can try to shoot it all down, and you have to defend it to your committee. Your committee basically is a bunch of professors in your department, and you actually have to go choose your committee. So you have to go ask people, would you like to be in my committee? Thank you, you'd like to be, and then they're in your committee. So at first you're asking them, but as soon as you ask them, they become your overlords. It's kind of a weird situation. You invite them to be on your committee, and then suddenly they're going to start telling you, and so they're your committee, and they give you this oral exam. When I did my oral exam, it was not on what I had planned to do. They had a different thing where you had to think about completely different research. So I was working on yeast stuff, and so I did a whole bunch of research into how flowering happens, the genetics of flowering, and I had to propose, if I was doing this, here's what I would do, here's all the experiments I would do, and then I had to go present this to the committee, and they had to shoot it down and try to find holes in what I had planned to do. And the point with something different was they figured that if I'm just going to talk about the yeast stuff, I could just get all the answers from my professor, and that makes it too easy. So I had to go find something else that he wasn't an expert in to see if I could come up with myself what it was to do. But either way, you have to pass some kind of an oral whatever. And so that's kind of the situation he's in. And then, by the way, at the end, you have to defend your dissertation in front of the committee as well. You give I gave an hour-long, it's pretty similar. You give an hour-long talk about your research over those years, and then anyone can come to your talk. My wife came and friends came, whatever. It comes to your talk, and then they dismiss everybody else except for your committee, and then they give you the oral, whatever. They try to shoot you down. And by the time you get to the defense of your dissertation, generally they don't let you do that unless they're pretty sure you're going to pass. If they think you're not ready, then they won't let you do it. So it's not like you're guaranteed to pass, but you're pretty close. But it's still super, super stressful. You stand in front of these people and they start asking, well, how come you didn't do this experiment? Why did you do this? What? And you have to like defend everything, So that's at the very end. But so here's Mitchison in front of his committee. What did his committee think he should be proposing to do? Pretty much, yeah. All these other centrosome stuff, different in vitro kind of experiments. He was looking at more of a sort of a more of in vivo, how things really work, and they didn't like that, so they almost, they basically would have thought, well, he's not a good scientist because he's not proposing valid kind of experiments and whatever, and almost flunked him. And of course, they have egg on their faces now.
Yeah, isn't the paper like one of the ending of this paragraph? It talks about how like none of those things were actually viable like experiments now that we like realize, and they're barely like starting now with this.
Yeah. So that was really fun. All right, so one of the things that was they noticed that was interesting was the number of microtubules would increase over a broad concentration range. So again, if you have like free tubulin dimers floating around, the more higher concentration of free tubulin dimers, you would expect that you have more and more polymerization because there's more free ones that can join the thread and make it longer. And if you had less, you'd have less. But he sees in a big different range of very high concentration, very low. You still get things growing, you still get things shrinking. So that was very interesting. And one of the other things he did was look at individual microtubules. What had other people done instead of looking at individual microtubules? Yeah. Just sort of the whole cell, or sort of an in vitro situation where you can sort of look at bulk tubules. One of the ways you can do that is with spectrophotometer. So you probably all use spectrophotometers, right? You shine the light through there, and it gives you a readout. And so if you have free tubulin like this versus they're all polymerized into tubules, this one blocks the light, and this one the light can kind of go through it better. So you can kind of tell if they're polymerized or not polymerized, kind of just by shining the light through there. And that's what other people had been doing. And it kind of gives you an average number, 90% polymerized or 10% polymerized or whatever you can get. But what you couldn't see there was some growing and some shrinking at the same time. And you can only see that when you look at individual microtubules. And you can kind of see those with like fluorescence microscopy, like the picture we have, they were growing and shrinking. No one had done that before, which was kind of surprising in hindsight, that was such a kind of easy thing to do, but no one had done it before. And so he starts discovering this whole, dynamic instability because he was looking at it in ways that people didn't used to look at it. Let's see. So then they came up with their model of the GTP cap here. We postulated the unusual kinetics of microtubules were due to two populations of polymers. Some that were growing had a cap of GTP tubulin, and others had lost the cap and were depolymerizing with GTP and exposed, and they call this dynamic instability. And then they kind of show in this figure number 6, kind of what they call the purpose here. These experiments suggested a purpose for dynamic instability that transcended the mere assembly of the polymer. So basically it shows that it can grow and shrink until it finds a sort of a target, and the target will stabilize it, and you get the tubules in the right place. And the ones that didn't hit the target would just shrink. So you end up with tubules, and we already talked about this the other day too. They don't have eyes, so they can't see where they're going to go. They just have to just grow and shrink randomly in different directions. But the ones that are supposed to hit this side of the cell where the little targets are, those are the ones that get stabilized. So they're exploring the interior until they find what they're looking for. And then subsequent work, this very last paragraph before the authors, many components that interact with microtubules have been identified since their 1984 papers on dynamic instability. They are fascinating proteins that bundle, sever, stabilize, and destabilize microtubules. So this is like what we talked about the other day with the actin. There's all these proteins that work with that. There's that great figure in the book, so go back and read that again. That shows the bundling proteins, the sequestering proteins, the severing proteins, all that. So the same thing, different proteins, but same kind of idea with microtubules. A vast array of motor proteins play roles in sliding materials on microtubules. One might have said that the study of microtubule dynamics in 1984 was premature because the entire cast of characters were unknown. Anyhow, what other comments do people have on the paper, on this article, this whatever, remembrance, nostalgia? They wrote this many years after the fact. Anyone like the papers? Was it fun to read? You know, a little bit of how stuff is done. When you read papers like these, peer-reviewed, published research articles, you're not going to hear about how someone nearly flunked their qualifying exam. That's not the kind of thing they're going to put in here. You get that from something like this. So I found this great website that has a whole bunch of these. So we're going to read some more for the cell cycle. Yeah.
I just have so who wrote this? Because this is obviously this person really admires, and then they just decided to write this summary of how the research had gone.
Yeah, obviously. I'm pretty sure that they wrote it. So really? Kirshner and Mitchison, yeah. Because the other ones, this one kind of used third person. You know, Mark did this and whatever did this. Some of the other ones, I believe, say I and we. So we got some more of these for cell cycle stuff, and I think they're more of a. I can't remember if they're, but yeah, they wrote their own. They wrote their own stuff. I just found this great website with a whole bunch of discoveries. I wish we had time to do a whole bunch more, because everything we ever talked about, there's someone that had discovered it and they wrote some. Well, most of them had written up something like this. And, you can kind of see what the process was. All right. Any other comments? All right. In that case, let us start talking about chapter 20, and then the schedule is that Wednesday, whatever we don't get to today, we'll kind of catch up and bring the review sheet. We'll try to do a little review on Wednesday as well for the test on Monday, and then we'll start cell cycle stuff on Friday. That gives everyone the weekend. But if you suddenly realize that, oh yeah, I do have airline reservations for Saturday and I'm not going to be here on Monday, then please let me know sooner, and rather not have to go all the way through the whole Thanksgiving break with people who haven't done that yet. Let's see here. So, yeah.
Is chapter 20 going to be on?
Yeah, the first half, the part we're talking about today. So chapter 20 is kind of, the first half is cytoskeleton, extracellular matrix kind of stuff, and the second half is sort of, to me it's a weird organization. I think there's some rationale for it, but it's kind of a weird, I don't know why this didn't take. Let's try it again. What's new? Well, no, I don't want to watch IT movies. I want to watch my iPad here. Get the little wheel going around. Anyway, the reason I put these together is the cytoskeleton's all the little ropes and cables inside the cell, and the extracellular matrix is all the stuff outside the cell. They kind of both are used to kind of support and give some structure to things. This is not cooperating here. Let me start over again. Extracellular matrix is the stuff that cells make, but it's not made of cells. So in your body, you've got cells, but your body's not entirely made of cells. Like your bones, there's cells in there, but it's not just pure cells. It's calcium deposits and stuff. So that's all extracellular matrix, all that stuff between your cells that's not made of cells, but it's made by your cells. Your hair is an example. Your hair is not alive. It's not made of cells, but obviously your cells make it, because where else is it coming from? Your fingernails are also not alive. They're not made of cells, but obviously cells make the stuff that makes your fingernails, and so it secretes it out, and so your hair and your fingernails. That's two examples. But even between our cells and different parts of the body have more or less matrix material. All right, we got this to work finally. That's reassuring. Tissues, basically organized groups of cells, they're all kind of working on a similar process. Notice the epithelial on the top, that's your outside skin cell. That looks like it's pretty pure cells, but underneath, those fibroblasts, those green ones, those are these weird-shaped fibroblast cells, but they're kind of floating around. All that gray stuff is the extracellular matrix that they've secreted, so they've made all this stuff. And so different layers of cells have more or less of it. And then you've got the muscle cells, and they're going in different directions. There's a cross-section. So the ones here are going that way, the ones underneath are coming out this way. And again, connective tissue underneath, you've got, again, sort of this gray stuff, which is like matrix, extracellular matrix material. These are just some interesting pictures, but I'm going to try to move on to here. Oh, so this talks a bit about plants. And again, it's nice to not diss plants. So on the plants, just a little bit on plants. So plants have cell walls. They don't have much of the same sort of extracellular matrix that animals do because they've got walls around their cells, which kind of do the same idea. Plant cell walls are made of a bunch of things, but mostly cellulose. So you've got to have, here's the membrane here on the bottom, and then you got to have, so I get this to work again. No, it's not working. Here's the membrane, and then you've got sort of the cell wall out here outside the membrane, and these are all cellulose. And cellulose, again, is a polymer of glucose, so it's basically a polysaccharide. But they're fibers of cellulose that kind of run in parallel directions. And so the plant actually lays down the cellulose in kind of parallel fibers. And this is how it controls which way the cell is going to grow. So it's like putting a belt on. If you put a belt on, you can't grow wider, but you can grow taller. So the ones on the top, they're kind of like belts going around the cell. The cell's not able to grow out because those belts are in the way. Instead, it can grow up and down. But if you put the belts on around the top, then it cannot grow up and down. Instead, it can grow out. So the plant will plan which way it wants the cells to grow and put the cellulose down accordingly. In order to put the cellulose down, cellulose is too big to create inside the cell and try to push it out through the cell membrane. So it has an enzyme, and the enzyme is in the cell membrane. That's this blue thing here, and it's basically the enzyme is putting glucoses together to make cellulose. So it's kind of getting made. So the enzyme is right in the membrane, so it's getting made, and then the enzyme moves, and so on the inside it's on microtubule tracks. So you've got these. So what the cell has to do is put its microtubules inside in exactly the same pattern it wants its cellulose outside. And then these enzymes just move down the tracks and then lay down the cellulose on the outside. And so however the tubules are on the inside, the cellulose follows. So here's the picture on the top. That's the cellulose on the outside, and then right underneath there's the microtubules, the same pattern, the same direction. So that's an ingenious way to do it. So you're just sort of creating it as you go, as the enzyme moves down the tracks. And that's mostly what the chapter talks as far as plants goes. There are different kinds of cells in plants, just like there are different kinds of cells in animals. Different types of cells. One you should know is the phloem and xylem. How many people are familiar with phloem and xylem? All right, what are they for? That's their transport. So now does this come out? I know botany has been sort of de-emphasized lately here. Does this come out in general bio?
Yeah.
All right, so that's good. I'm reassured by that. So again, xylem is the little water pipes, and phloem is the sugar pipes. So you make sugar in the leaves by photosynthesis and then take it down to the rest of the plant through the phloem pipes, and the water comes from the roots through the xylem pipes. And the way to remember which one is which, there might be more than one way. The way I remember which one is which is phloem starts with a pH, and the sugar, that's the food for the plant, and the sugar also starts with a pH. So there's your food, and there's your phloem. pH, phloem, I think it's something like that, phloem and food. And that leaves your water to be xylem, so I don't know. There's no good X thing for xylem, but that's how I remember what the phloem does. So you have specialized tubes just to do that. The xylem, again, for water, These are the water tubes. The xylem are dead cells. So end to end, they're stacked up. Both of these are 1 cell wide. So it's 1 cell wide, just stacked up end to end, and then there's holes in the walls so that it makes it sort of a continuous tube. The xylem are dead, so the cells have died, and all that's left is the walls. And the phloem is still alive. So the phloem are still alive. Now, to make the phloem more efficient, a lot of the stuff is actually removed from the cell. A lot of those organelles are kind of removed. I think even the nucleus in some cases might be removed so that more can be better piped. So there are like these companion cells next door that they basically feed these cells all the stuff they need because they don't have their own stuff anymore. At least some of their nuclei and stuff. So these cells kind of feed these cells so that there's a better. They don't have to be blocked with all kinds of organelles, and then you have a nice pipe going up and down. And then there's lots of other kinds of plant cells as well. But at least if we kind of just get that much as far as plants go. And then we'll kind of go back to animals for the rest of this chapter. Here's bones. If you've had physiology, you probably know more about bones than I do, but it's mostly sort of calcium deposits, and then there are like these cells that are sort of scattered through there, but it's a lot of extracellular matrix inside of bones. And then collagen. So there are a few extracellular matrix proteins everybody ought to know. Collagen is one of them. So, and keratin is another one. How do you spell keratin? So this is the one that's in your fingernails and hair, but it's also other places in your body as well. So it can be in your skin, between your skins and all that stuff. Collagen is also underneath your skin layer. It kind of gives the elasticity and stuff to your skin. So these are both proteins. And unlike a lot of proteins that kind of fold up in these globs, these are more rope-like. They're kind of spread out like this. Collagen is involved in bone formation as well. So if you don't have proper collagen, you can't get proper bone formation. This is what when we had Julia back in 2010 and long before she was born, when she 6 months before, she was only 3 months along, the doctors did the ultrasound and said the bones weren't right. They could tell, even the baby's only like this big, and they could already tell the bones weren't right. And they tried to actually sequence the DNA to find what the mutation was, and they never found it. But it's probably it was a collagen defect. And so if you have a collagen defect, your bones can't form right. And there's different collagen defects, and I've learned all this from our experience. Depending on where the mutation is and what it does. And some screws up collagen more than others. And so if you have a milder defect, you can still get what's called, it's a osteogenesis imperfecta. And it's, bones aren't right. And a couple of years after that, there's a volunteer couple here at SVU named the Joneses. I don't know if I took the clock off because it wasn't right. Does anyone remember the Joneses? They're both music people. They've been gone. They've been gone for 3 or 4 years now. One of them played the piano, one played the organ, and Sister Jones, they did a fireside one night, and she played the piano, and then she got up and said, "I've got these special rings on my fingers because they bend backwards because I've got bone issues and they bend backwards, and so I've got to put these rings on when I play so that my fingers actually stay straight and don't just kind of flip backwards." So I went and talked to her later, and she's like, "Yeah, well, I've got this disease, and it's called osteogenesis imperfecta, and you've probably never heard of it before." And I said, "Oh, yes, I have." And of course, she had a milder version of it, but it makes your bones bad, so there's the rings, and it also makes your eyes discolored, and she's a little short, and that could be for any reason. Lots of people are different heights, but that's one of the things. And I asked her, do you mind if I talked about this in class? And she said no, and that's why I'm talking about it. I don't normally talk about people's diseases without their permission, but she said no. And so I told her we had a daughter that had this, and she had a very severe case, and she was born with broken bones by C-section. They wouldn't let her have a normal birth because that would cause more breakage, and her ribs didn't quite develop normally, so that's why she had problem breathing, and she was in the NICU and at UVA for a couple of days, and we were incredibly fortunate to have her for that long, because we didn't even know we even get that much. So collagen, basically, it's one long strand, and then you braid them together. So triple-stranded collagen, and braid them all together. So then we take that braid and make a whole bunch more, and they take that and make a whole bunch more. So it's like building up. You have one single one, you braid it, and then a whole bunch of those makes the next one, a whole bunch of those makes the next one, and there's your collagen. And collagen is outside the cells, and it's also too big to make inside the cells. So again, it's kind of secreted. You can make the protein, and then you have to kind of secrete that and then kind of form the larger bundles of it outside the cell. And the fibroblasts are the cells that go around, and they actually crawl. So these cells crawl around, and they kind of like spiders leaving their web behind. They kind of crawl around, leaving collagen in their wake, kind of behind them. And they can crawl around different directions to make the collagen in the right place. We're out of time. We'll talk about vitamin C. We'll talk about vitamin C starting on Wednesday. Let's see. Let me just see where we are on this.